Okay, so let's talk about clot retraction. That is going to be the fourth step in our process here. And this generally happens within 20 to 60 minutes. So clot retraction. So again, usually happens 20 to 60 minutes after that clot is formed. And how it does that is it actually will squeeze serum from the clot when it joins the edges of that broken vessel. And you can think about joining the edges kind of like stitches do if you would have a wound that would be big enough for stitches. Now this whole process requires really a lot of platelets. So you can think of someone who didn't have a lot of platelets, this would be a difficult thing to do. So the less platelets, the less clot retraction. The fifth and final step is what we call clot dissolution. Okay, so clot dissolution happens fairly shortly after clot formation, and what it does is it allows blood flow to be reestablished and for a permanent repair to take place. And sometimes it'll repair with a scar, sometimes not. There are just a short sequence of steps that occur with clot dissolution. So they involve activators and inhibitors. So the first thing that we're going to see is plasminogen. And plasminogen is going to be converted to plasmin by a number of activators. And these activators are made in the endothelium, the liver, and the kidneys. So then what happens is the plasmin digests the fibrin strands and some of the clotting factors. And that's how we get rid of our clot and it heals. So some of the plasminogen activators that we're talking about are TPA, and that is tissue type plasminogen activator, also something we can give someone, administer in cases of stroke and things like that. It is basically a clot buster. And the other one is UPA, urokinase type plasminogen activator. So just know that these two are clot busters. They're going to bust up those clots.